You may see more clones across the valley in the near future.
That's because an Ivy Tech professor is teaching a class how to clone plants from stem cells. I met with the instructor to see how it's done. Cloning may seem like science fiction, but it's something that happens all the time with plants. Ivy Tech Life and Physical Science Assistant Professor Mark Flynn teaches that to his students as part of a biotechnology program. They clone African violets using stem cells.
Plants have stem cells just the same way that people and everything else has stem cells. It's just they're a little easier to control with plants and a little easier to grow from a sample.
Students remove a portion of the leaf on the plant near the stem or near stemmatic tissue. The tissue is placed in a medium containing nutrients to feed and stimulate the plant. The process to grow the cells to a leafy plant can take many weeks.
They cut a small section out of that we're going to transplant into the shoot media to grow the top half of the plant. Then after about 6 weeks, when it's grown leaves, they'll transplant that into the root media to grow the roots. That takes another 6 weeks, and then they wind up with a small plant that we're going to transplant into soil.
There's a specific, practical, and local reason that Flynn combines tissue science and biotechnology in his class.
Something that everyone is exposed to in Indiana is corn and soybeans growing out in the field. And a lot of people don't understand that that's actually a biotechnology company that produces those plants in a very similar way to how we produce these African violets here.
The growing plants are kept in a greenhouse at room temperature with high humidity. They get about 12 hours of light through special light bulbs that simulate sunshine. Out of the past 6 years, students have used only 2 African violet plants to produce hundreds of clones.